Welcome everyone to the second in our new FBI Cyber series. I'm going to talk today about some older published work from my lab, some brand new data hot off the press. We were just in the midst of collecting some of these data when we had to shut down operations for the virus. So I've been busy trying to make some slides based on some data that Stephanie and Parva just sent me. So the data I'm going to show you, the project I'm going to show you has been a thread that's kind of unified my lab from day one. It's pretty much the very first project I started when I became independent, started my lab. And so it's a series of research that's near and dear to my heart. Let's see. So this is now quite a famous paper, a seminal paper by Alison Goat when she was at UW in collaboration with other well-known people like Laura Beirut. And this paper was published really just after I'd started my own lab. And it struck me as really surprising. I think most people in the field as being really surprising. And what this paper reported and why it was so surprising is that they'd identified, Alison and her colleagues had identified allelic variation in nicotinic receptor subunits that contributed to vulnerability to tobacco dependence. And that in itself may not sound surprising. Nicotine acts at nicotinic receptors. So of course, you would think if there's genetics involved in vulnerability, it would be in the primary targets that respond to nicotine. But what was so surprising was actually the subunits in which these the alleles existed, the allelic variation. The subunit genes were the alpha 5, alpha 3, and beta 4 subunits. And why that's surprising is because these subunits are not very prevalent in the brain, not expressed densely. And in the few sites in which they are expressed, those sites have not typically been associated with nicotine addiction. The alpha 3, alpha 5, and beta 4 are pretty much considered ganglionic, or were considered ganglionic nicotinic receptors, as opposed to the alpha 4 and beta 2 subtypes, which are where they're considered the high affinity nicotinic receptors. Most people who work in nicotine addiction, or any aspect of nicotine's actions have really focused their attention on alpha 4 and beta 2. So the fact that Alison and colleagues found that this reproducible marked signal in terms of genetic vulnerability to addiction was in these so-called ganglionics was very strange. And that effect is being replicated by many groups at this stage. These were a series of papers published not long after Alison published her paper, showing that the very same alleles that seem to influence tobacco dependence vulnerability play a critical role in vulnerability to a whole host of smoking-related diseases, lung cancer, cardiovascular disease, et cetera, et cetera. And the reason for that, the most probable reason for this association is because you get these diseases like cardiovascular disease, lung cancer, because you smoke. So these alleles are really important for vulnerability to tobacco dependence. And yet, at that stage, we knew remarkably little about these subunits. Very few people were working on them because people really didn't care about them because they were, as I say, considered ganglionic and baroque and not very important. And it turns out they are remarkably interesting and very important. So I'd mentioned that in the brain, they're not really in regions that we were at that stage considered or thinking about in the context of addiction. In the context of the alpha 5 nicotinic receptor subunit, alleles in which have been reproducibly associated with tobacco dependence vulnerability, you see that alpha 5 nicotinic receptor subunit transcripts are really not very prevalent in the brain, and when they are expressed, as I said, they're in these regions that typically we hadn't thought of in the context of addiction, like the medial habenula to interpeduncular nucleus pathway, the nucleus tractus solitarius, NTS, nucleus of the solitary tract. The habenula-IPN, we know very little about what it actually does, and yet it's one of the major two so-called convection systems in the brain, mechanisms by which information flows from the forebrain, the limbic system down into the midbrain, into these kind of actuator sites that influence different types of behaviors. The other major convection system, of course, is the medial forebrain bundle. That's well known for its role in reward. So if you activate the medial forebrain bundle, you recruit the ventral tegmental area, the nucleus accumbens, et cetera, and that's considered a major reward system in the brain. Yet the habenula to interpeduncular nucleus is equally as involved in terms of connections between the forebrain and midbrain, yet we know really nothing about what it does, what its role is. And I'll get back onto that in a little bit. And alpha-5 nicotinic receptors are really densely expressed in that circuit. And in addition, as I said, in the NTS, and the NTS is a hindbrain nucleus best known for its role in regulating appetite. It's also the site in which the vagus delivers information from the periphery, so interoceptive information, if you will, and a whole host of other information into the NTS, and from the NTS it gets routed into various other regions of the brain. And so these sites, these two circuits, if you will, or regions of the brain, are highly enriched in alpha-5 nicotinic receptors. And so we began to explore the role for nicotinic receptor signaling in these two parts of the brain. I will say, in fact, we'd already begun to explore the role for nicotinic receptor signaling in these areas because I'd already been interested in alpha-5. It was only really when Allison's alpha-5 nicotinic receptor subunits, it was only really when Allison's paper was published that I realized that they may actually be important, and it was really worth focusing a lot on what those subunits may be doing as opposed to other nicotinic receptor subunits. And so this work I'm going to show you is long since published now. It was led by Christy Fowler while she was a postdoc in my lab. And what Christy found was after she set up an intravenous self-administration procedure that could be used for mice, so we can measure the reinforcing properties of mice. She examined the reinforcing properties in wild-type mice and mice in which the alpha-5 subunit gene had been null mutated, so they can't express alpha-5 subunits. And when she looked at self-administration behavior in the animals, it's shown there on the bottom right, she found in wild types that we see the classic inverted U-shaped dose response curve, which is when we increase the unit dose of nicotine available for self-administration, wild types begin to consume more and more of the drug as the unit dose is thought to be more reinforcing. But as you increase the dose even further, it begins to become aversive. So there's a mix between reward and aversion, and the animal moderates its intake as a consequence, and intake begins to go down. It's getting more bang for the buck, if you will, at those higher unit doses. And it wants to avoid any aversion from those higher doses. But when you look at the alpha-5 knockout mice, what we found is that the lower doses, at lower doses, self-administration was unaltered between wild type and knockouts. These are the doses that we think are maximally rewarding with minimal aversive properties. But as you increase the unit dose even further, we found that the alpha-5 knockouts continued to take nicotine at doses that the wild types avoided. So from that, we suspected that what was going on with alpha-5 and related nicotinic receptors is that maybe they're not regulating nicotine reward per se, maybe they're regulating nicotine aversion, and they're involved in nicotine avoidance behaviors at these higher doses. And if you don't have these alpha-5 subunits, your detection of those aversive effects of nicotine is compromised, and as a consequence, you use more of the drug. That's exactly what we found to be the case in data I'm not going to show you. We reported a lot of it in Christie's paper and a few follow-up papers. But what I'm going to show you is the areas in the brain in which alpha-5 nicotinic receptors are likely involved to regulate those noxious aversive properties of nicotine. And as I said, the alpha-5 subunit transcript is enriched in the habenula-IPN pathway. So that was one of the first sites we examined. We took our knockout mice, we cloned the missing alpha-5 subunit into a lentivirus to be able to re-express it, and we targeted that lentivirus into the habenula of either wild type or knockout alpha-5 knockout mice. The idea here was that we could rescue, if you will, the otherwise missing alpha-5 subunits in the brains of the mice. And when we did that and looked at self-administration behavior in the animals, what you can see in the bottom left is, or the bottom right corner, the left panel, is in wild type and knockout animals treated with control virus, this lenti control panel, that the alpha-5 knockouts in red again consumed much more nicotine than the wild type animals, and that effect was most notable at the highest unit dose of nicotine. And on the bottom right, if we virally re-express alpha-5 subunits specifically in the habenula-IPN pathway, we rescue that effect. The animals now consume amounts of nicotine very similar to wild type animals. So from that, we hypothesized that nicotine acts in the habenula-IPN pathway through a mechanism that requires alpha-5 containing nicotinic receptors. And when nicotine stimulates this pathway, that triggers some kind of aversion or avoidance signal that results in the animal limiting or moderating its consumption. And if you don't have that alpha-5 mediated event in the circuit, animals will continue to use nicotine over and above what you would see in an animal that has normal alpha-5 signaling. And in fact, this finding, this interpretation has now been extended into humans, and that's exactly what occurs in people who carry alleles in which alpha-5 subunits don't function as well as those individuals who don't carry those alleles. It turns out that people with those risk alleles are less sensitive to nicotine aversion or noxious properties of nicotine, and consequently they use more of the drug. And we think that's what's responsible for these alleles resulting in greater levels of nicotine dependence. It's not because the drug is more rewarding, it's because it's less aversive and you're less likely to avoid it. Ultimately, that results in consuming quantities of the drug that remodel the brain in all those very interesting ways that result in compulsive behaviors. Okay, so that was Christy's contribution to this project. And then a graduate student in the lab stepped in. It's Louis Tuesta. I should mention Christy is an associate professor at UC Irvine now. She's done really well. Louis has just taken a faculty position at the University of Miami, his hometown. But when Louis was in my lab, he was interested in exploring the role for this other population of alpha-5 expressing neurons I'd mentioned in the NTS, the nucleus of the solitary tract. We're curious if this population of cells that express alpha-5 nicotinic receptor subunits may also play a role in regulating nicotine intake. So we're curious what those cells were, which class of receptor in the, or cell in the NTS may express alpha-5 nicotinic receptor subunits, and are those cells involved in regulating consumption of the drug. So we've looked at this in various ways. One very straightforward way was simply injecting animals with nicotine to activate, presumably activate the cells that express nicotinic receptors in this part of the brain, kill the animals and stain for Nicotinic activity. So we look for cells that are activated and then stain to see if we can identify the phenotype of those cells. And we already know that one of the major populations of cells in the NTS are catecholaminergic in nature. The NTS, in addition to the locus coeruleus, are the two major regions that provide ascending norepinephrinergic input into the midbrain. So these norepinephrinergic cells are a major subpopulation of neurons in the NTS. So we thought, of course, that nicotine is activating any cells in this part of the brain. And if any cells are going to express alpha-5 subunits, it's going to be these tyrosine hydroxylase positive norepinephrinergic cells. And we're very surprised to find that actually when we stained for tyrosine hydroxylase, we found that the phosphoramino activity that was triggered in response to nicotine injection did not co-localize with TH positive cells. So that meant that this well-known, very well-studied population of cells in the NTS are not those that are likely activated by nicotine. And there's another mystery population, if you will. So we went hunting to figure out what they may be. And it turns out that there's a group of cells in the NTS that express or produce a neuropeptide called glucagon-like peptide 1 or GLP-1. And when we co-stained FOS with a GLP-1 antibody, we found that those GLP-1 cells were in fact activated by nicotine. I should say they're not the only population of cells in that part of the brain that are activated by nicotine. There are other cells that are GLP-1 and TH immunonegative. We don't know what those cells are. We did look using other antibodies, and we haven't been successful in figuring out what they are currently, although we are using single cell sequencing. So more recently, so I'm optimistic we will figure out those interesting populations down in that part of the brain. However, what I can say is that the GLP population of neurons down there are activated by nicotine. And when we looked at an alpha-5 GFP reporter mouse, we found that indeed, if we stain for GLP-1, that those very same cells seem to be GFP positive. So the GLP-1 cells appear to produce alpha-5 nicotinic receptor subunits. So we thought, that's interesting. Here's another part of the brain that expresses alpha-5. There's a discrete population of neurons that expresses those receptors. Are those specific cells, those GLP-1 producing cells in the NTS, involved in regulating nicotine intake? It turns out that the GLP-1 is produced from a gene called GCG, or glucagon. So GLP-1 is produced from pre-pro-glucagon, which is the parental peptide, if you will. And it turns out that there are various Cre drivers that will target these cells. For example, pre-pro-glucagon-1 or FOX2 Cre. All these lines of mice will express Cre recombinase specifically in those GLP-1 producing cells in the NTS. So we took advantage of that to target these GLP-1 producing cells in the NTS that are activated by nicotine and that express alpha-5 subunits. And we either turn them on or turn them off selectively using DREADS. And what we found, this is just one example here, minimize that. If we chemogenetically stimulate specifically the GLP-1 producing cells in the hindbrain, in other words, what we think nicotine is doing, so mimicking the actions of nicotine, we found that animals self-administered less nicotine. And conversely, data I'm not showing you, if we chemogenetically inhibited these cells, animals took a lot more nicotine. So we think that activation of these cells is kind of like activation of the habenula. In other words, alpha-5 nicotinic receptors in the habenula-IPN, when they're activated, it triggers an aversion signal, a avoidance signal, animals use less nicotine. And it looks like the same thing is happening in this part of the brain too. If you activate these GLP-1 neurons to express alpha-5, it seems that animals will use less nicotine. So presumably there's some kind of aversion or avoidance signal that's being triggered also by activation of this other population of cells in the hindbrain. So how does this work? What happens when you activate these GLP-1 neurons in the NTS such that animals use less nicotine? So again, I'm summarizing a lot of data here in just one slide, but this is already published. It's published a year or two ago from the lab. It turns out that when you look to see where GLP-1 receptors are in the brain, the densest concentration by far is in the interpeduncular nucleus, the IPN. This very site I just spoke about that has very dense concentrations of alpha-5 nicotinic receptor subunits. So we thought, well, here's a very nice mechanism. Perhaps the NTS is projecting to the interpeduncular nucleus, that these GLP-1 neurons are projecting down to the IPN. They're secreting GLP-1 to act on the dense concentration of receptors that's in the IPN. And essentially, it's moderating the effects of nicotine on the habenula to IPN circuit. So we tested that hypothesis. We tested whether GLP-1 neurons project to the IPN and regulate the activity of IPN neurons. And we've done that in a few different ways. Just here's a little bit of data using cell physiology from the SAGO in the lab. And we expressed channel rhodopsin specifically in the GLP-1 neurons in the hindbrain, and then we optically stimulated terminals in the IPN, and we did that. We found that we could detect an optically evoked excitatory current in those IPN neurons. Interestingly, we only saw that excitatory current if we stimulated at 20 hertz and not 1 hertz. Why is that important? Well, typically you have to stimulate above 20 hertz or above to depolarize or to trigger release of these dense core vesicles in neurons that contain neuropeptide. And you typically can't accomplish that if you stimulate at 1 hertz. So by using a stimulation protocol that we think will trigger release in neuropeptides, it's only then that we see an increase in the activity of IPN neurons, or an excitatory current release in IPN neurons. So we think that there's GLP-1 receptors throughout the habenula-IPN circuit. They're able to respond to GLP-1 that's released from these GLP-1 neurons that arise in the hindbrain. And we think that that's likely important in controlling nicotine intake. But can we test that hypothesis? And we've done that in a few different ways. The first is we used an shRNA, which is pretty effective at knocking down GLP-1 receptors. We delivered that shRNA via an AAV into the habenula-IPN circuit to knock down GLP-1 receptors in that circuit and presumably attenuate, reduce the ability of that circuit to respond to GLP-1 from the hindbrain. And we did that. Sure enough, our animals self-administered a lot more nicotine. Conversely, if we deliver GLP-1 receptor agonist, in this case it's called exendin, into either the habenula or the interpeduncular nucleus, there's a dramatic decrease in nicotine self-administration behavior. It's quite profound. The animals look otherwise normal. They'll respond and consume food normally. They'll move around the cage normally. But as soon as they self-administer a single infusion of nicotine, they almost immediately avoid the lever for the rest of the session. Occasionally you get an animal that will take a second infusion, but that's quite rare. And that's shown in the bottom right there. Exendin being infused, in this case, into the interpeduncular nucleus. And we already know that GLP-1 signals through cyclic AMP. It's positively coupled to adenylate cyclase. And if we block cyclic AMP signaling whilst we stimulate GLP-1 receptor transmission in the IPN, we can basically completely block the effects of this GLP-1 receptor agonist on nicotine intake. So all that to say that we think that these hindbrain GLP-1 neurons are projecting to the habenula-IPN circuit. They're stimulating or enhancing the sensitivity of that circuit, we think, to nicotine. And when that occurs, animals use less nicotine. And if you disrupt either the activity of the habenula-IPN or this input from the hindbrain, this GLP-1 input from the hindbrain, animals use a lot more nicotine. So we think by following on Allison's original observation in humans, identifying polymorphisms in alpha-5 nicotinic receptors, we've stumbled upon this entire circuitry in the brain that's controlled by alpha-5 receptors that seems to regulate nicotine avoidance behaviors. And so we've asked a few questions subsequently. One of those questions was, well, how does GLP-1 actually act on this circuit in the brain? Because the effects are profound. And if we can understand how GLP-1 is acting, we may be able to design interesting therapeutics, effective therapeutics for smoking cessation. You may say, well, we've used a GLP-1 receptor agonist in the previous study I just showed you. Why not just put that into smokers? It turns out that that is currently in clinical trials. It's gone into a phase 2 clinical trial just about a year ago after we published our work. And so we're waiting to see if there's an efficacy signal. But one of the drawbacks of that compound is it's also used for the treatment of diabetes. So GLP-1, in addition to being produced in the brain, is also produced in the gut. It's secreted in response to gastric distension. So when you have a meal, GLP-1 is secreted, and it goes and acts the pancreas to enhance insulin secretion. So we thought it's great to have something that will boost GLP-1 signaling, but it'd be nice to have something that can act like GLP-1, but not modify insulin per se, because that's something we'd like to avoid, particularly as smokers are known to have issues with blood sugar regulation. So we wanted to see how GLP-1 acts in the habenula and hopefully tap into other mechanisms by which we can modulate habenula-IPN activity. So this work I'm going to show you was, it was led primarily by Alex. It was his thesis work, but he had a lot of help from people in the lab like Stephanie, Molly, and Masago, many others. But what Alex asked really was, okay, what do we know about GLP-1 signaling, and can we begin to flesh out the cellular machineries, if you will, through which GLP-1 is acting on the habenula, but potentially identify other mechanisms that we can manipulate the habenula via that machinery. So it turns out that there's a little bit known already about how GLP-1 acts, or the canonical signaling cascades, if you will, from its actions in the pancreas. So if you activate GLP-1 receptors, as I already mentioned, they're positively coupled to adenylyl cyclase, so you get an increase in cyclic AMP. That's taught to recruit various cyclic AMP responsive protein kinases that will phosphorylate various things, one of which is beta-catenin, in a non-canonical site that will stabilize beta-catenin, which in turn will translocate to the nucleus, where it will bind with a transcription factor called TCF7L2 and trigger a whole host of genes to be transcribed. So we thought, well, maybe this very same machinery is also in the brain, perhaps in the habenula-IPN circuit, and this machinery is responsive to GLP-1 and controls nicotine intake. So that was our hypothesis. So as a first step, we wanted to go to the very last step in this machinery and start working backwards. So what would happen if we knocked out TCF, knocked out beta-catenin, knocked out the kinases that are involved in phosphorylating beta-catenin, et cetera, and begin to flesh out the machinery through which GLP-1 is acting. So as a first step, we wanted to knock out TCF7L2. Well, first of all, we want to see if TCF7L2 is even in the brain and in the areas where we think GLP-1 is acting. And if so, can we manipulate TCF signaling in those brain regions? And lo and behold, when we looked in the Allen Brain Atlas, it turned out that perhaps the densest concentration of transcripts for TCF7L2 in the brain is actually in the medial habenula. And if you immunostain for TCF, that's shown in the bottom left in green, you see that there's dense protein concentrations for TCF in the medial habenula. You can see there in red, that's actually a chat-cree TD tomato mouse. That's a long way of saying that this mouse reports where cholinergic neurons in the habenula are. This is just a good way of orientating ourselves. So we knew we were looking at the medial habenula, that's those red cells. And hopefully you can see that there's a lot of green where we see red, although it doesn't always overlap. There's lots of other types of cells in medial habenula. All that to say that we think there's a lot of mRNA, a lot of gene transcripts, and a lot of protein for TCF TCF7L2 in the habenula. So that's good. That's where we think that GLP-1 is acting on those habenula cells that project out into the interpedunculair nucleus. So, okay, the transcription factor is there. Is it transcriptionally active? Well, there's TCF7L2 reporter mice available. It turns out if you just take these mice and kill them and stain for transcription factor activity, in this case LAC-C, so it's a beta-gal stain, you can see even under basal conditions that the habenula is blue. What does that mean? It means likely that TCF7L2 is active in the habenula. So the transcripts are there, the protein is there, and it's functional in the medial habenula. So that was all good news because it meant that the transcription factor that we think GLP-1 is signaling through is actually in the site in which we think GLP-1 is acting on. So with that in mind, we want to manipulate TCF7L2 and see what would happen to nicotine self-administration behavior. The first thing we tried to do a few years ago was get a hold of a TCF7L2 knockout mouse. It turns out that they die soon after birth. And the reason they die is because when they're no longer in utero and a mother is providing food, and soon after birth, they have to burn or use stored energy in their liver. In other words, they have to produce glucose themselves until they can feed. It turns out that the mutant animals are not able to do that. And they starve to death essentially in the first few hours after birth, unfortunately. That meant we couldn't use them. But we thought, well, rats are much more robust. They're able to do without food for much longer periods of time than mice. So if we made a knockout rat, perhaps it would be able to get through this early stage soon after birth until it's able to feed. And the mother, so it can feed from the mother before it can suckle, and it won't die in that period before it's able to consume calories again from the mother. That was our rationale for making a rat as opposed to using het mice that look like they are viable, but the knockouts are not. And I thought I heard a question there. Okay. All right, so we use zinc finger technology, this is in collaboration with Aaron Gertz, to target TCF7L2, specifically the beta-catenin binding site. And we were successful in doing so. So we generated a TCF7L2 mutant animal, a mutant rat. We checked the habenula-IPN and the rest of the brain, and it looks like it's intact. Structurally, it looks like it's intact, and a whole bunch of data I'm not going to show you. Functionally, it looks like it's intact. So we looked at behavior in these animals, and one of the first things we did is look at intravenous self-administration, and it turns out that the knockout animals consume copious amounts of nicotine, really a lot of nicotine. This looks very much like the alpha-5 nicotinic receptor subunit knockout mice I showed you. It also looks at like GLP-1 receptor subunit or GLP-1 receptor knockout mice, in data I haven't shown you. And it's consistent with, again, this circuit, this habenula-IPN-NTS circuit being important in regulating nicotine intake. However, this is a whole body constitutive mutant animal. We have manipulated TCF specifically in the habenula. So of course, that was something that we wanted to do. We've done this in various ways, one of which is definitely designed a really nice guide RNA to be able to cleave TCF7L2 DNA. And we then express selectively Cas9 in the habenula and express this guide RNA to cleave TCF7L2. We confirmed that we could do that. We could do it effectively and selectively in the habenula in mice. And when we cleaved TCF7L2 in the habenula, what we found is that the cleaved animals consumed, again, just remarkably high levels of nicotine. These are really huge levels, hugely increased doses and amounts of nicotine that are consumed by these mutant animals. It's shown in the middle in terms of the number of infusions they earn. And if you do a quick calculation by multiplying the infusions by the dose, get the actual amount of nicotine the animal consumes at each dose. And you can see it is a dramatic increase in consumption. So all this to say that we think that GLP-1 signals through TCF7L2 in the habenula-IPN circuit. And TCF7L2 is really critically important in regulating nicotine intake. But TCF7L2 is a transcription factor. How is it working? What is it doing to control nicotine intake? We spent a long time trying to understand that. And one of the things we did was to take the habenula and some other control regions in these animals and deep sequence the habenula, IPN, hippocampus in either wild type, TCF7L2 wild type, or the mutant animals, and look to see which genes are dysregulated. So which genes are under the control of this transcription factor. And perhaps one or more of those genes will explain why the animals take more nicotine. And we thought, well, this is probably pretty easy. TCF7L2 is likely transcriptionally controlling the expression of nicotinic receptor subunits, including alpha-5. And if you don't have TCF working properly, you don't have nicotinic receptors, and essentially you've made an alpha-5 knockout animal. It turns out that was not the case. Alpha-5 and other nicotinic receptor subunits are expressed at similar levels in a mutant as wild-type animals. So that surprised me, because I thought that was a pretty parsimonious, simple explanation for everything that was going on. So it meant there were other mechanisms. We looked for a long time until we just actually stepped back and looked to see what the gene expression profiles, the data were telling us. And if you take the genes that are differentially downregulated in the habenula of the knockout or mutant animals compared to wild types, it seems that there's a preponderance, a concentration of genes that are under the control of cyclic AMP that are downregulated in the habenula of the mutant animals. And from that, we thought, well, maybe TCF is somehow controlling genes that regulate cyclic AMP signaling. So maybe cyclic AMP is not working properly in the habenula. So we looked at that in various ways, one of which is we developed this ex vivo transcription factor reporter approach that we're now trying to apply in vivo. And it's essentially the same type of approach you'd use in cultured cells, where you can use luciferase or destabilized GFP to look at the activity of transcription factor. And we expressed that reporter in the habenula of either wild type or mutant animals, and then stimulate cyclic AMP signaling in the habenula. We did that by basically putting on a GLP-1 receptor agonist, which acts through cyclic AMP. And what we found is in wild type animals, we got a beautiful increase in cyclic AMP signaling in the habenula, and that effect was pretty much completely abolished or absent in the mutant animals. So that means that TCF702 is controlling cyclic AMP signaling in the habenula, and if you don't have functional TCF in the habenula, you don't have functional cyclic AMP signaling. Why might that be important to the regulation of nicotine intake? It turns out it's been known for a long time. This is classic work from Rick Huganier and Paul Greengard, showing that cyclic AMP responsive kinases play an absolutely critical role in regulating the function of nicotinic receptors. And very specifically, you need these cyclic AMP mediated kinases in order for a nicotinic receptor that's being desensitized because it's being activated by acetylcholine nicotine to return to an active state. So without cyclic AMP dependent kinases, if you desensitize a nicotinic receptor, it's pretty much going to stay desensitized. So here's a mechanism whereby TCF can control cyclic AMP and thereby control nicotinic receptor function in the habenula. And it turns out that's exactly what's happening. So Masago in the lab was able to isolate nicotinic receptor currents in habenula neurons in wild type and knockout animals. He found that if he delivered nicotine in high frequencies, that's shown the bottom right in that kind of blue outline. Oh, sorry, you can probably see my mouse. Here we go. Here we delivered nicotine with high frequency. We desensitized these nicotinic receptors. If you then stop delivering nicotine at high frequency, you can see that in wild type animals, nicotinic receptor function recovered. If you boosted cyclic AMP signaling, it really didn't do anything in those in nicotinic receptor currents in those animals. In the knockouts, what we found is if under basal conditions, nicotinic receptors were just fine, but if you desensitize them, they did not return to normal levels of functioning for as long as we recorded them. However, if we enhance cyclic AMP signaling, we could rescue that deficit. So we think we have an interesting mechanism whereby GLP-1 enhances cyclic AMP. We think it enhances this process here, recovery of nicotinic receptors from desensitization. That's why animals are more sensitive to nicotine. If you knock out TCF7L2, you compromise this process. Cyclic AMP dynamics are compromised. As a consequence, nicotinic receptors are compromised. And as a consequence of that, animals consume a lot more nicotine. So that's all captured here. So as I'm running, I want to get through all this new data. I'm going to keep on going. So basically, nicotinic receptors, we think, are under the control of GLP-1 and TCF7L2 in the habenula. If you activate these nicotinic receptors in the habenula-IPN pathway, that basically triggers nicotine avoidance behaviors. But if GLP-1 and or TCF are not functioning properly, cyclic AMP signaling is not functioning properly. Nicotinic receptors are more likely to remain in a desensitized state in response to nicotine. Consequently, the habenula is less sensitive to nicotine and avoidance behaviors are not as marked. It's an interesting mechanism by which drug intake can be controlled. It's the mirror image, if you will, of the reward circuitries that many of us are interested in too. Here it's an aversion circuit. Rather than enhanced reward, this is diminished aversion, diminished avoidance, and consequently, the motivational properties of nicotine are enhanced. I'm just going to show you one other little piece of data. It's something that we stumbled upon pretty much by accident. But I told you that we looked at gene expression profiles in the habenula of the TCF7L2 wild type and knockout animals. I showed you that if you looked at the differentially downregulated genes in the knockouts, that cyclic AMP responsive genes or transcripts look like they were impacted in the mutant animals. It turns out if you look at the differentially upregulated genes, the TCF mutant animals in the habenula, pretty much every pathway that we found to be dysregulated was involved to some degree in glucose handling, glucose metabolism, glucose in some manner. And you may say, well, I'm looking here and I see Alzheimer's disease as one of the pathways or genes involved in Alzheimer's that's dysregulated in the habenula. It turns out if you look even in that particular pathway, they're all glucose relevant genes that trigger or that throw up Alzheimer's as a pathway. Alzheimer's disease is a pathway that may be dysregulated. This was really surprising. I was not aware of, and there is not any literature, or wasn't any literature, on a role for the habenula in regulating any aspect of glucose metabolism, glucose regulation, but it was very interesting from the published literature. It turns out that TCF7L2, allelic variation TCF7L2, is or TCF7L2 is the gene that's been most implicated in genetic vulnerability to diabetes. It's been replicated over and over and over again. So you've got alleles that modulate TCF activity. It'll influence whether you develop type 2 diabetes. It turns out that one of the other major factors that will influence whether you develop type 2 diabetes is whether you're a smoker. And we thought that this is just too much of a coincidence for not to mean something. Perhaps diabetes in smokers is coming from some action of nicotine on the habenula that's controlled by TCF7L2. Maybe nicotine is able to influence blood glucose in some manner, and it does it by this action of nicotine on the habenula involving TCF7L2. Or maybe all this is just coincidence, but we thought it was worth looking. So the first thing we did is actually look to see what would happen if we gave animals doses of nicotine that activate the habenula. What would happen to blood glucose? So we delivered intravenous doses of nicotine. We functionally imaged the animal's brain using fMRI. We identified a dose of nicotine that will activate the habenula. Turns out it's on the descending portion of the dose response curve. In other words, one of these doses where if you have an alpha-5 mutant animal, they will consume a lot more of the drug. So that wasn't surprising, but it was nice to see that we could verify that effect, that these higher doses that wild type animals avoid, but mutants don't, were the doses that activate the habenula. But when we let animals self-administer those doses to activate the habenula, what we found is that it caused really a striking increase in blood glucose, which is really surprising. And in a whole host of experiments, I'm not going to show you except for one, if we manipulate TCF7L2 in the habenula, we can completely block this effect. In other words, that nicotine stimulates the habenula, and we think by stimulating the habenula, that triggers through some mechanism an increase in blood glucose. And if you abolish TCF signaling in this part of the brain, you can attenuate that response. How on earth does that occur? How's the habenula, nicotine acting on the habenula, able to increase blood glucose? Well, through a bunch of experiments, we mapped out a circuit whereby it seems that the habenula via the autonomic nervous system, the sympathetic nervous system, is communicating polysynaptically with the pancreas. Basically, we turn the habenula on, you're able to turn on pancreatic alpha cells in the pancreas, and in doing so, you secrete glucagon that acts in the liver and blood glucose goes up. So that's a lot to remember. All you need to know is that nicotine acts in the pancreas, and in doing so, blood glucose is increased. And what we found is if we let animals have a lot of nicotine for a long period of time and looked at the pancreas and looked at their blood glucose, that wild-type animals developed what looked like diabetes-type dysregulation in blood glucose homeostasis, because this entire circuit becomes completely dysregulated. So nicotine, by repeatedly stimulating the habenula, will dysregulate the pancreas and blood glucose, and that effect is not detected in these TCF7L2 mutant animals. So we think that the diabetes that we see in smokers is actually not because of pancreatic dysregulation per se, or at least that's not the origin of the disease. Instead, it's coming from habenula dysregulation. So this is the same diagram I already showed you. Nicotinic receptors regulating habenula responsiveness. It turns out when you activate the habenula, you're going to get not just nicotine avoidance, but a big increase in blood glucose. And if you do that frequently enough, you get a whole host of adaptations in the circuit from the habenula all the way down to the pancreas, such that when you stop using nicotine, the pancreas is completely dysregulated, and you end up with this really marked perturbation in the ability to regulate blood glucose. Your blood glucose levels are elevated and you have what looks like type 2 diabetes, but the diabetes is originating in the brain. So if that's the case in smokers, what else is originating in the brain? So there's a whole host of smoking-related diseases that look like they're associated with altered autonomic system function. Cardiovascular diseases, for example, hypertension. Are those diseases caused by dysregulation of habenula and other sites in the brain that are under the control of nicotine? It's something we're really interested in and hopefully going to begin to look at again soon. Okay, so I'm going to see if I can move through this pretty quickly, because that brings me to Stephanie's project in the lab, one of Stephanie's project in the lab, and she's kind of picked up the baton here. So we've kind of taken a step back recently, and that is, well, not recently, something we've been thinking about for a long period of time, and that is the question of what is it that the habenula-IPN pathway is actually doing? It's not there to respond to nicotine, presumably, or maybe it is, as you'll see in a few minutes, but presumably it's there for some fundamental reason. It's quite a heavily conserved pathway in terms of mammalian conservation, mammalian species. It's across pretty much any species that you care to take a look at. Reptiles have a very well developed habenula, as I say, pretty much all mammals. So it's heavily conserved across evolution. So it must be doing something. And the question is, what is it doing? And so Stephanie's been asking, a broader question: what might the habenula be doing with regard to and its involvement, its implication in regulating these actions of nicotine? So we've taken a look in the literature, and there's not that much known actually outside the role for the habenula and IPN pathway in the context of nicotine and nicotine's actions, in terms of what that same circuit might be doing in the context of other behaviors, other physiological processes. But if you look far back enough, you can find that there are some papers that provide these tantalizing hints as to what this and related components of the habenula-IPN pathway might be doing. It turns out that the habenula, medial habenula, receives input almost exclusively, in fact, I'd say exclusively, from the septum. And if you lesion the septum, the posterior septum, which is the site that provides input to the habenula specifically, the habenula, the intranuclear nucleus, or the NTS, you alter an animal's ability to detect bitter tastings. So things that are bitter that otherwise animals don't like the taste of and would normally avoid. And there's a very well worked out circuitry, reasonably well worked out, I should say, circuitry that responds to bitter tastes and transduces that signal in the brain. And our own Ivan Duruju is playing really a critical role in mapping out some of the circuits that are involved in not just bitter, but sweet taste processing, and the systems that are involved in regulating behavior in response to consumption of these different types of tastings. So essentially, you consume these tastings, there's activation of taste receptors in the mouth. That signal is transduced into the NTS, the nucleus to solitary tract. It goes either monosynaptically or likely parallel polysynaptic circuits as well via the gustatory thalamus, and ultimately ends up in the insular cortex, the gustatory cortex, and gets routed back down to the NTS. And so it seems that the habenula-IPN pathway circuit is part of this broader circuitry that's involved in regulating sensitivity to or transmission of bitter tastes and once you consume bitter tastings. So we think that perhaps we've tapped into a circuitry that's there naturally to respond to what are considered noxious substances. So plant-derived alkaloids are known to be bitter, and quite often they're poisonous. So we think that this is a system that developed for animals to avoid potentially poisonous and toxic substances, hence this system being involved in aversion. And nicotine is either directly or indirectly recruiting this circuit, and that's why it's triggering avoidance behaviors. And I should say that previously in data I'm not showing you, we've already shown that the insula plays a key role in regulating nicotine intake. So we think that this broader circuitry is involved in that process. And we think that that's actually really interesting, because nicotine is, as I just mentioned, a plant-derived alkaloid that's actually bitter. It tastes bitter. And in fact, pretty much any other addictive drug you care to look at is also bitter tasting, and most of them are plant-derived. Even the artificial addictive drugs, if you will, the synthetic addictive drugs, like oxycodone or semi-synthetic addictive drugs, have the same type of structure that renders them bitter tasting. So they recruit this bitter taste circuitry. And it's kind of interesting when you look in terms of other bitter relevant effects in smokers. Not only is nicotine bitter, but it turns out if you've been smoking for a long period of time, you may know that your taste or sense of your perception of food has been altered. And it turns out that the reason for that is because you get a very specific deficit in your ability to taste the bitter. Sweet taste and other forms of taste are unaltered, but the more you use nicotine, which is bitter tasting, the less able you are to detect bitterness. And another interesting tidbit, it turns out allelic variation in the receptors that respond to bitter tastings, like other plant-derived alkaloids, in this case they're called T2Rs, allelic variation in T2R genes has been associated with increased vulnerability to smoking behaviors. So from that, we came up with a hypothesis. Perhaps this septum to habenula to IPN to NTS and broader circuitry involved in the insula, thalamus, mouth, and perhaps other components is involved in detecting noxious substances. It's there to respond to these substances, like nicotine or other alkaloids. And then if that's the case, nicotine is able to recruit the circuit in probably two ways. It's able to recruit the circuit directly because when you consume nicotine, it's able to penetrate directly to the brain and activate nicotinic receptors that are expressed in this circuitry. Or indirectly, because when you consume nicotine, it's in the mouth and there's these sensory processes, these taste receptors that may be able to detect nicotine and transduce that sensory property into the brain, into the circuitry via the traditional sensory conduction into the NTS. So a direct or indirect mechanism. So we thought that we've already demonstrated that disruption of nicotinic receptor actions in the habenula-IPN will lead to an increase in intake. So if we disrupt this direct action of nicotine, it causes an increase in consumption of the drug. What would happen if we block this indirect action? Say if nicotine is bitter, it is being detected in the mouth. If we disrupted an animal's or a human's ability to be able to detect that bitter property of nicotine, of the drug, would that attenuate the overall recruitment of this aversion circuit and would greater amounts of the drug being consumed, would greater amounts of nicotine be consumed? So how might nicotine or how are other bitter tastings detected in the mouth? This is pretty well worked out. You've got taste receptors in the mouth. They're expressed on the tongue. They're expressed within taste buds that are able to detect different modalities of flavor. And those different taste buds components have different types of receptors. And one type of receptor is the so-called T2Rs, or the bitter taste receptors. So there's a specific class of receptor that exists to detect bitter alkaloids, a whole system in the mouth that's able to detect these types of chemicals. So we thought if we could disrupt taste signaling in the mouth, maybe what we'd see is less detection of nicotine and greater consumption of the drug as a consequence. So how do these bitter taste receptors actually signal? So you see here all these different types of taste receptors. One thing that's common to all of them, and it's just the bitter taste receptors shown here in a bit more detail, is that they're G protein coupled receptors. They signal through G proteins, but the G protein that they use is a special, interesting kind of G protein. It's called a-gustducin. So it has these taste receptors, these T2R receptors, bitter receptors, have their own dedicated G protein, which is advantageous from an experimental perspective because it means if we target, delete, or cleave gustducin, we disrupt the ability of bitter tastings to operate. So that's something to keep in mind. So we first asked, does nicotine activate the bitter taste receptor system? And these are experiments carried out by Stephanie and Pervin in the lab. These are ongoing experiments, but we're actually pretty excited about them. The first thing we did was to culture STC cells. These are cells that are derived from the intestine. They constitutively express taste receptors in the machinery to respond to bitter tastings. So they naturally have this machinery. We screened shRNAs against alpha-gustducin, if you remember. That's the GNAT3 gene. If you remember, that's the G protein through which these taste receptors signal. These are the control cells, the control SH shown here. And what you can see here is all these different shRNAs that we screen, nice knockdown of alpha-gustducin. So we're able to pretty effectively cleave alpha-gustducin expression with a number of different shRNAs. We selected 2 of these shRNAs and looked to see, just to confirm that they're working, So in STC cells that express bitter taste receptors, we exposed them to the most bitter substance known to man, denatonium. And what you can see is we get nice activation of these bitter taste receptors reflected by an increase in intracellular calcium. And if we knock down a-gustducin in these cells with 2 different shRNAs, in both cases, we markedly attenuate, in fact, abolish the effect of denatonium on these cells. So that means our shRNAs are working. Strikingly, if we put nicotine onto these cells, we could get similar activation, and we markedly attenuated the ability of nicotine to activate these cells if we knock down a-gustducin. And to my mind, this is kind of a remarkable finding. It means that in addition to nicotinic receptors, the traditional way that we know that nicotine acts to stimulate cells, most notably neurons, there may be a whole other host of receptors that are able to detect the actions of nicotine because it's an alkaloid. These receptors may respond directly to nicotine, or they may respond indirectly because nicotine is acting through nicotinic receptors, but those nicotinic receptors are interacting with taste receptors via GNAT3. And in either case, it seems that in STC cells, you need GNAT3 in order to respond fully to nicotine. So this taste machinery looks like it's important for these taste-sensitive cells to respond to this alkaloid. It turns out it's not just nicotine that shows this effect. We found that these STC cells that express taste receptors also respond to oxycodone, cocaine, caffeine, and some other alkaloids. So there's a whole parallel system that will respond to addictive drugs, not just the primary site of action, like in the case of cocaine, the dopamine transporter, oxycodone, opioid receptors, nicotine, nicotinic receptors, because by virtue of the structure and the factor alkaloids, you have these alkaloid sensing taste receptors that will also respond to drugs like nicotine. So it's interesting, how do these alkaloids actually work? So Stephanie has gone to see what would happen in terms of brain activation if you deliver these various different alkaloids that we're interested in. Just shown 2 that Stephanie included in her experiments, nicotine or oxycodone. So she delivered these to animals, and she used whole brain CISFOS-mapping to identify circuits in the brain that may be activated by nicotine, oxycodone, etc. We are very interested in the circuits that we've been interested in for a number of years, those I've been talking about, the habenula-IPN-NTS, etc., to see if they're being activated by these addictive drugs and other alkaloids. And also, is that activation sensitive to these taste receptors? If we disrupt taste receptor signaling, either pharmacologically with T2R receptor antagonists, or by cleaving GNAT3 or knocking out GNAT3, will that influence the ability of these addictive drugs to activate the brain in these sites that are responsive to bitter tastings? So Stephanie injected animals with these drugs and other bitter tastings that are not addictive drugs, cleared the brain. This is a brain from Alex Smith, a separate project, but it's the same approach. We clear the brain, stain for FOS in a whole brain manner, and then look for regions that are activated. And what Stephanie found was across the board for all these different alkaloids, the NTS was remarkably sensitive, as were other regions of the brain, but the NTS really stood out. And that is not a surprise, of course. I already mentioned a few minutes ago that the NTS is known to be a core component of the circuitries that respond to bitter tastings. When they're detected outside the brain, that information gets routed to the brain via the NTS, so perhaps not a surprise. And this is, if I can show you this quickly, this is just one of Stephanie's brains with the clearing approach. And what you can see here is nice FOS immunoactivity down here in the NTS. I'm going to stop this for a second, if I can. Let me just, well, I just want to point this out because it's going to become important in a couple of minutes. This little structure here that you can see in the ventricle is the choroid plexus. And I pointed out because you see it's almost contiguous with the areas that are around the NTS. But as you deliver animals with various alkaloids, including nicotine, oxycodone, you can see that the NTS, which is right by the ventricle, right adjacent to the choroid plexus, is activated. So very interesting. What's even more interesting is if you pharmacologically block bitter taste receptors using a combination of T2R receptor antagonists, you can markedly attenuate the ability of nicotine to activate the NTS. And personally, I think this is absolutely striking. So nicotine is turning on this major circuit in the brain that I've shown you earlier, plays a critical role in regulating nicotine consumption. And if you block this whole other separate population of taste receptors, presumably in the mouth and elsewhere, you're able to attenuate the actions of nicotine in the brain. And just to confirm that these antagonists are acting at taste receptors, if you look at the same effect in a GNAT3 knockout animal, the effect is even more profound. You almost completely abolish the ability of nicotine to activate the NTS. So you need these taste receptor signaling mechanisms in place in order to be able to respond to nicotine, or at least for the NTS to be able to respond to nicotine. Is this important? We looked at this in various ways. One is we let animals drink nicotine. So the idea here is nicotine is bitter. We detect it in the mouth. It's going to signal indirectly into the NTS, saying that something bad has happened to some bitter tastant. And if you can block that effect, presumably animals will consume more of the drug. It's exactly what we see. So animals have the choice to be able to consume water or nicotine adulterated water solution. And you can see in wild-type animals, or sorry, control animals, they don't consume too much of the nicotine, but if you block taste receptor signaling, that effect is, or that avoidance is markedly diminished. Animals consume a lot more. You get the same effect in the GNAT knockouts. I'll show you that in a second. First of all, we confirmed that the GNATs are sensitive to bitter tastants, or sorry, the GNAT knockouts are less sensitive to bitter tastants. This is this denatonium compound I mentioned previously. What you can see is in wild types, they will begin to really markedly avoid water that's been adulterated with denatonium, and that effect is diminished in the alpha or the GNAT3 mutant animals, and you get precisely the same effect in those animals if you let them consume nicotine. So it seems that the detection of nicotine orally, presumably, controls consumption of the drug, and if you block the detection, the bitter taste properties of nicotine, you consume a lot more of the drug. What we were really surprised about is it seems that if you block taste receptors by giving these taste receptor antagonists systemically, so, or sorry, if you give nicotine systemically, not letting the animal drink the drug, but give nicotine by a subcutaneous injection, you get exactly the same effect with taste receptor antagonist. So here we looked at condition-place preference. Here animals are paired to one side with nicotine, one with saline. In this case, nicotine is given systemically. It's not into the mouth, it's given subcutaneously. And if we block taste receptors, we actually reverse an aversion effect, an aversive response to nicotine, such that the drug looks like it's rewarding. That means taste receptors are controlling response to nicotine, but the taste receptors in the mouth are likely not involved because we're given nicotine by a subcutaneous injection. We get exactly the same effect in the GNAT3 knockouts. They also show enhanced reward to nicotine, but nicotine has been given subcutaneously. It's not being delivered orally. So taste receptors are regulating this effect, but not taste receptors in the mouth. If we look at self-administration of the drug, exactly the same thing. If we block taste receptor signaling via subcutaneous injection of taste receptor antagonists, you get increased consumption of the drug. And we wanted to look at that in the GNAT3 knockouts, but it turns out that we couldn't train them to self-administer for various reasons. Turns out they're very anxious. So we'd like to do that, but it's not technically feasible. But you can see the effect is pretty clear if we pharmacologically block taste2 receptors. So this means that bitter taste signaling seems to play a critical role in regulating the motivational properties of nicotine, but it's likely not bitter taste signaling in the mouth, bitter taste signaling occurring elsewhere. Where are these taste receptors located that are regulating this response to nicotine? Where are they? And so I'm going to go through a lot of data in like 1 minute, but we've been hunting around to see where this may occur. One of the experiments we did is single cell sequencing of the NTS, the idea being if we could identify really which populations of cells in the NTS respond to nicotine, that may tell us where the information is coming from, presumably from the periphery. These taste receptors expressed in some organs in the periphery, if we can figure out which cells in the NTS respond to these bitter tastants, that will tell us which organs we should look at. And what we found, and it's difficult to see from here, but what we found is the cells that are most responsive to nicotine were not actually neurons in the NTS. They were choroid plexus cells. They were the very cells I just pointed out in Stephanie's iDisco video, which are these cells that are located in the ventricles that are not thought to really do anything in the context of behavior. Their job is to produce CSF, and that was largely what they were taught to do. Turns out if we do single cell sequencing in the septum, and the animals respond to nicotine. What you can see here is control animals. These are choroid plexus cells. These are animals in response to nicotine. This is by far the biggest change in gene expression that we see is in the choroid plexus. So we think that the choroid plexus may be super responsive to nicotine, the septum, NTS, and other regions. And it turns out that's important. Why? We looked into the literature. It turns out choroid plexus cells actually express taste receptors in GNAT3. In other words, it's not just the mouth that can potentially taste nicotine. The brain can potentially taste nicotine and can taste nicotine via these taste receptors that are expressed in the choroid plexus. So can nicotine act on the choroid plexus? So these are new data where we cultured human choroid cells and we treated them with nicotine. And we looked at calcium response. Hopefully you can see that. There's a beautiful increase in calcium response. So nicotine is able to stimulate choroid plexus cells, in this case, cultured human choroid plexus cells. And if you block taste receptor signaling in these cells using TAS2R antagonists, you can see that the effect of nicotine is really dramatically abolished. What you can see here is the effect of nicotine is bimodal. You get this really marked increase in calcium response, and then it seems that calcium signaling or the responses, if anything, is depleted. And if you block taste receptor signaling, you attenuate that effect. We've now done this experiment in vivo using fiber photometry, where we express GCAMP in the choroid plexus, in the fourth ventricle. Given animals nicotine, we get an increase in calcium in choroid plexus cells in the fourth ventricle, and then you get this depletion in calcium signal, if you will, or attenuation of the signal. So we think nicotine is turning these cells on quite robustly, and if anything, turning them off afterwards is a consequence because it's if anything, overstimulated them. Is this important, and are these choroid plexus cells important in regulating nicotine intake and this bitter taste response? Well, Stephanie has gone and designed a guide RNA against GNAT3, validated it, and have targeted the choroid plexus and confirmed that she can knock down GNAT3 in the choroid plexus. And when she does that, we get exactly the same behaviors that we see in the GNAT3 knockout animals. Their avoidance of nicotine is incredibly diminished. If anything, the effect in the choroid plexus deleted animals is more striking than the whole body knockouts. Animals just do not avoid nicotine. They consume a lot of the drug when it's available for drinking. And if we look at CPP, conditioned place preference, we can basically reverse what is either a non-rewarding or reversible effect of nicotine and turn the drug into a rewarding, that dose into a rewarding dose by deleting the ability or cleaving GNAT3 and attenuating the ability of the choroid to detect nicotine. Anyhow, I'm a couple of minutes over. I apologize. I'm setting a very bad example for the rest of this seminar series. Anyhow, I think the take-home message here is that we've tapped into a circuit that seems to be critical in regulating detection of noxious alkaloids. And we think that this system is probably critical for the addictive properties of nicotine and other addictive drugs. And I want to finish by thanking everyone in the lab, particularly Stephanie and Purva, who are in the midst of this taste receptor project, and everyone else has helped. And happy to answer any questions. Hey, Paul.
How you doing?
Good.
Yeah. So I have a quick question. So very, very nice talk. I love the it's a very complete story that you have there. But one thing I picked up on earlier in your talk was that the alpha-5 knockout, that you pointed out that the mice were not responding to a low dose of nicotine. But then in the TCF7L2Z mice, they did seem to be responding to a lower concentration of nicotine.
Yeah, absolutely.
I was wondering if that meant that there were other proteins involved in the response to nicotine. Yeah,
likely there are. It also probably reflects the fact that TCF is not just in the habenula-IPN. The alpha-5 nicotinic receptors are pretty restricted in terms of their expression profile, whereas TCF is also in the thalamus, it's in the PAG, it's in some other regions that may play a role in regulating nicotine intake. So it's probably a combination of both.
So do you think they're still all involved in aversion? So it seemed like maybe what happened is you just shifted the aversion curve even more, or made it less prominent in the transcription factor knockout.
Yeah, that's exactly what I think is happening. And a lot of data I didn't show you speak to exactly that. It looks like it's not reward, it's aversion that's being impacted.
Thanks.
Paul, this is Alison.
Hey, Alison.
The TCF7L2 polymorphisms that influence risk for type 2 diabetes.
Yep.
Do they also have an impact on smoking? I think they changed the level of expression, but I can't remember which way round it is. And
yeah.
so.
Yeah, we've looked at that, Alison. It's really interesting. So it turns out that the TCF7L2 mutations that are associated with diabetes are likely gain of function mutations. And so if you look in humans that carry those mutations, what you see is that they're less likely to smoke. If you gain TCF, those mutations are, it's really quite a marked effect. And then if you look at the interaction between smoking and TCF mutations, it's quite profound. If you covary smoking in individuals who've got TCF7L2 risk alleles for diabetes, in other words, you dial out the effects of smoking, the risk associated with TCF7L2 is markedly diminished. So what that means is if you have a TCF mutation, these mutations, you're less likely to smoke. But if you do smoke, you're much more likely to get diabetes. Really, really interesting kind of gene by environment interaction. Anyhow, I'm sorry, Alex. I think we're 8 minutes past the hour.
I'm going to send you an email about taste receptors.
Okay. Right. Thanks, everyone.